Next Generation Sequencing, NGS. Next Generation Sequencing, NGS, also known as high-throughput sequencing, is a revolutionary technology that has transformed the field of genomics and genetics. It is a powerful and cost-effective method for analyzing and sequencing DNA, RNA, and other biomolecules at a large scale. NGS has significantly advanced our understanding of genetic variation, gene expression, and the underlying mechanisms of various diseases. Unlike traditional Sanger sequencing, which could only sequence a single DNA fragment at a time, NGS can simultaneously analyze millions of DNA fragments in parallel. This massive parallelization allows for faster and more efficient sequencing, making it feasible to study entire genomes and transcriptomes in a single experiment. The basic workflow of NGS involves several key steps. 1. Library preparation. DNA or RNA samples are fragmented and adapters are added to the ends of the fragments. These adapters contain sequences that allow the fragments to bind to the sequencing platform. 2. Sequencing. The prepared DNA or RNA fragments are loaded onto the NGS instrument, and the sequencing reaction is initiated. The instrument reads the sequences of the fragments in parallel, generating massive amounts of short reads. 3. Data analysis. The raw sequencing data is processed and analyzed to reconstruct the original DNA or RNA sequences. Bioinformatics tools and algorithms are used to align the short reads to a reference genome or transcriptome, identify genetic variants, and determine gene expression levels. NGS has numerous applications in various fields of research, including 1. Genomic research. NGS enables whole genome sequencing, allowing the study of genetic variations, disease-causing mutations, and population genetics. 2. Transcriptomics. RNA sequencing, RNA-seq, using NGS is used to investigate gene expression levels, alternative splicing events, and the discovery of non-coding RNAs. 3. Epigenomics. NGS can identify DNA methylation patterns and histone modifications, providing insights into gene regulation and epigenetic changes in different cellular contexts. 4. Metagenomics. By sequencing entire microbial communities directly from environmental samples, NGS enables the study of complex microbiomes and their impact on ecosystems and human health. 5. Clinical applications. NGS has revolutionized personalized medicine by allowing the identification of genetic mutations associated with various diseases, which can aid in diagnosis, treatment selection, and patient management. Over the years, NGS technologies have continued to evolve, with improvements in sequencing accuracy, read length, and throughput, making it even more versatile and valuable for research and diagnostic purposes. As a result, NGS has become an indispensable tool in modern biology and medicine, paving the way for groundbreaking discoveries and advancements in the life sciences. Don't forget to like, share, and subscribe for more exciting content on the latest technology trends and innovations. Thanks for watching.